Hello and welcome. At first glance, the gut and the eye seem far removed from each other within the structure of our anatomy. But now more than ever, researchers are looking into the link between the gut microbiome and ocular disease. This is the second interview in our two-part series on the microbiome of the eye. I'm Stephanie Stewart-Bailey, the museum specialist for the Truhlsen-Marmor Museum of the Eye. Together with Jenny Benjamin, the director of the museum, we are excited to welcome today's speakers from the National Eye Institute. Before I introduce them to you, I want to share a little background about this research. The human gut houses up to 70% of the immune system. Within the intestinal tract live up to 100 trillion organisms, an ecosystem linked to both innate and adaptive immune function in animal models. Propelled by advances in high throughput gene sequencing, evidence is suggesting that the gut's microbiome impact a range of immune-mediated diseases, including multiple sclerosis, type 1 diabetes, rheumatoid arthritis, and cardiovascular disease. In ophthalmology, recent research is looking into the link between the gut microbiome and the pathogens of ocular disease. So in today's common usage of microbiome, it refers to the community of bacteria, viruses, fungi, and yeast, plus all their genetic material that live in or on our body. So just how complex is the gut microbiome? An estimated 1,000 species of bacteria alone live in the human digestive system, with 10 times the number of genes in the human genome. Only a few of these microbes are well understood. So it's as if in a vast orchestra, only a handful of instruments can actually be heard. But these notes are early, and they might lead to new therapeutic paradigms where the gut microbiome plays a role in treating eye disease. So throughout this program, we will be discussing cutting-edge research that is still in development. All right. Well, we are just so excited to have the both of you join us today in conversation. Thank you so much for joining us and taking the time to explain your research to us. So it's my understanding that the two of you work together, but I'm wondering, Rachel, can you give us a brief introduction and tell us a little bit about where you work and what you do?
So both of us are in the Laboratory of Immunology in the National Eye Institute in Bethesda, Maryland. Reiko is a senior staff scientist in the lab, and I'm the section head.
And Reiko, can you tell us a little bit about what you do?
Right, so I do the experiment, planning of the project with Rachel, and also supervise some junior people to work together to produce those data.
Brilliant, brilliant. Today, we are focusing on uveitis, which occurs in the middle layer of the eyeball, and it gets inflamed, red, or swollen. This particular layer, called the uvea, has many blood vessels that nourish the eye. Uveitis can damage vital eye tissues, leading to permanent vision loss. Inflammation disease is when the immune system attacks the body's own tissues, resulting in inflammation or the swelling of tissues, as you see in this picture. Inflammation is part of the body's defense mechanism and plays a role in healing processes. For example, when the body detects an intruder, it launches a biological response to try to remove it. The attacker could be a foreign body, such as a thorn, an irritant, or a pathogen. Pathogens include bacteria, viruses, and other organisms which cause infections. So then sometimes the body mistakenly perceives its own cells or tissues as harmful. This reaction can lead to autoimmune diseases such as type 1 diabetes.
So in our case of uveitis, there is autoreactive T cells that can recognize the uveatogenic antigen. And if those T cells can see these antigen in somewhere in the body, it can get activated and start the immune process, like producing cytokines or some other molecule that cause inflammation. And also they can also recruit other cell types like leukocytes, neutrophils, other immune cells to even promote the inflammation itself.
Exactly, that's what happens. Everything starts with that T cell that has a misdirected thinking that it is attacking foreign tissue when it's actually self tissue.
Right. Okay, so as we just talked about, we developed the spontaneous model of autoimmune uveitis in our laboratory. And that mouse carries the retina-specific T cells. And it actually has a much higher frequency than normal wild type mice. So those T cells are more abundant in body of those mice, entire body. And what we think is happening in this mouse to have spontaneous disease is these T cells actually circulating throughout the body can, by chance, encounter the antigen, which is produced by gut microbiota. And we think it's a mimic antigen, which is similar to the retinal antigen, but not exactly the host protein. It's from the bacteria. And so the presentation of this mimic antigen in the gut to this retina-specific T cells get activated in the intestine and start to migrate through the circulation. And only the activated cells can cross the blood retinal barrier to get inside the eye to cause the inflammation. So in this case, these T cells are already activated in the gut, so they could pass through the blood retinal barrier to trigger uveitis by recruiting other inflammatory T cells. So that's this model says from 1 through 4 steps.
So I would add to this that this is actually has been a conundrum of immune privilege, because the eye is very protected from the immune system. And any kind of an inflammatory event is going to result in irreversible loss of sight. And so to the extent possible, the body separates the eye and the brain from the immune system. We know that in the brain, this has been kind of broadening and changing. In the eye, I would say not yet. We still do not see that, like they saw in the brain, that there are actually lymphatics draining the eye. It is thought that the eye does not have active lymphatic drainage as long as it's intact. Once you harm it, puncture it, it all leaks onto the conjunctiva, then you drain to the regular lymph nodes. But the healthy eye is very protected. It's called immune privilege. And one of the manifestations of that is that cells cannot freely traffic in and out unless they have been activated previously in the periphery, and they are now like metastatic tumor cells. They can cross blood tissue barriers because they break down the walls of the blood vessels actively by secreting matrix degrading enzymes, and then they can actually go anywhere. And so that's what we know from the immunization-induced model. And that's what we surmise is happening in the spontaneous model in order to induce disease, except, and this is of course an important point, that we have not actually taken a cell in the eye and proven that it came from the gut. This is ongoing work right now.
And is all of this in animal models still, or is any of it in human?
No, this is still experimental.
Can you talk a little bit about how you are trying to do that research and prove that a cell has been in the gut and found in the eye?
This is a research that is now ongoing. We have not done it yet.
I see. Okay.
You do it usually by labeling the T cells in the gut and then looking for them in the eye. Kind of a no-brainer. But it's a heck of a lot more complicated than it sounds when you say it.
Absolutely.
It's a whole different cup of tea. There are so many caveats and so many things that you have to be careful of, and pitfalls and things that can interfere. It's always a very low frequency of cells that you're talking about. So proving that this cell actually is there by design and not that it somehow got stuck in the vasculature but hasn't actually come out yet is necessary. And that's why we have these technologies of imaging looking at those labeled cells coming out into the retina. Well, let me put it facetiously. The eye has an inside and an outside, and the twain shall never meet. Seriously, whatever happens inside the eye is totally separate from what happens on the ocular surface. The sclera is very impermeable to stuff. It's even hard to get medication as eye drops. If it's a larger molecule, it just doesn't go in. So the ocular immune privilege is really something very, very basic and strong. And the only way to get into the healthy eye is either by physical trauma or by having an activated cell stick to the vasculature and actively break down the vessel wall and get in there. So this is the distinction between the inside and the outside of the eye that people really tend to disregard. And it's very basic in eye research that there's people, it's almost like having a specialization, medical specialization in the left nostril versus the right nostril, right? To bring it to absurdity. But really the responses inside the eye and the responses on the surface of the eye are totally different and governed by different rules.
Mm-hmm. What are some of those rules for the inside versus the outside?
So the inside has immune privilege. It is devoid of microorganisms and devoid of lymphoid cells. Mm-hmm. The outside is a mucosal site exposed to the environment. It has all the elements of a mucosal immune site, and it knows very well how to deal with bacteria because it has to do it all the time. Mm-hmm. But whatever happens on the surface of the eye very rarely affects what's going on in the eye.
Gotcha. And in our last conversation with Tony St. Ledger and Rob from UPMC, we were talking mostly about the exterior of the eye and the microbes that were found on that. And we did kind of like an interview of like, meet your microbes, and we talked about some of them. So here in the conversation with you, we're talking more about the internal and what is possible to be triggered or travel up to the interior of the eye.
I don't know if you know how we induce uveitis in experimental animals. We immunize them with an antigen that we extract from the retina, and we mix it up into an emulsion with mycobacteria. And actually that mycobacteria, per Janeway, who was one of the fathers of immunology, that is the immunologist's dirty little secret, the adjuvant, without which nothing works, and which is necessary in order to get not just uveitis, but a slew of other tissue-specific autoimmune diseases, of which uveitis is one. When you take a tissue antigen that resides in a particular tissue and not elsewhere, because otherwise it's a systemic disease like lupus, but brain, eye, adrenals, testes, other glands, thyroid, you can immunize with that antigen without complete front adjuvant, and not much happens. But if you throw in that complete front adjuvant, which is a suspension of mycobacteria in oil, all of a sudden the immune response goes into an inflammatory pathway because they need to chew up those mycobacteria. And anything that is in that same mix is getting responded to in a way that is tissue destructive. Hmm. So we sort of we're wondering, right around the same time, Reiko developed a spontaneous uveitis model where we cloned the unique T-cell antigen receptor that recognizes the retina. You know, every T-cell has its own, has a receptor that is unique to a particular protein. And when we immunize these mice, they develop T-cells specific to retina. But we were able to clone this T-cell receptor and engineer transgenic mice with it. And those mice, lo and behold, got 100% spontaneous disease by 2 months of age. And no adjuvant. So where is that bacterial signal coming from? So it was kind of a no-brainer to look at the gut at that point. Reiko, you want to expand on it?
Also, the gut has the commensal bacteria, most of it in the body, and next will be probably skin. So we started to look into the gut. And the first easy experiment to try was to deplete the gut microbiota to see whether the spontaneous uveitis is gone or lowered. And we did get that result about 10 years ago already by treating the mice with the antibiotic cocktail of broad spectrum antibiotics. And so mice are drinking those antibiotics daily to lose the gut bacteria. And those mice at the end develop spontaneous disease, but still a much lower disease score. So
okay. What about future therapeutic approaches? Do you see anything on the horizon there?
So actually there are analysis being done of human microbiome. And some people have already tried to put human microbiome into mice. And many of these were studies that were not really controlled very well, but it does seem that in humans with uveitis, there is a consortium of pro-inflammatory bacteria that can make the mouse that is subsequently immunized for uveitis. This is not in the spontaneous model, sicker. So actually this brings us to an important point that bacteria don't give just one signal. Adjuvant normally gives a signal that sends the cells into a type of pro-inflammatory tizzy, but it's not specific to a particular antigen. You can enhance response to any antigen with an adjuvant. But in this case, because the protein that is being targeted in the uveitis is shut up tightly in the healthy eye, these cells actually have to be not only excited, but also retina specific, because there is that important step of the cell getting into the eye. Unless it sees its antigen, it just goes back to sleep. So, and I'm sure that activated T cells, like the one that I showed you extravasating into the retina, which does happen to be a uvidogenic T cell, but activated T cells go into the eye as a matter of course, because that's what they do. Activated T cells are tissue seeking, and wherever they find a place to stick, they go out of the circulation. And so entry of even uvidogenic T cells into the eye is kind of random. They don't know where they're going. They're just swimming along happily with the bloodstream because that's what they normally do. And they get stuck in the small blood vessels, in the lung, in the liver, in the you know what. And as some of them get stuck in the retina because the retina is very heavily vascularized. And when they extravasate there into the tissue and they see their antigen, they say, uh-oh, this is something I need to respond to. And then they reactivate and they produce cytokines and they recruit all the nasty leukocytes that Reiko was talking about. And it's a self-amplifying inflammatory process that culminates in what we know as uveitis. Yeah, I think that what I would like to point out is how understudied the entire field of ocular immunology is. People are all rushing to do brain research, but they forget that if you are blind, well, you kind of lose a lot of quality of life, and it's very important to have good eyesight. And in biology, usually the deadlier diseases, the more money is poured into it, and the more attention is given to it. Because being blind or semi-blind or out of vision does not kill you, the eye kind of suffers a little bit of neglect as a stepchild. And I think that should change. Especially the eye immunology, probably more so than other disciplines like regenerative medicine. That they get more exposure. But inflammatory eye disease takes its toll. Well, uveitis, the classical uveitis that we're talking about in humans, is 10 to 15% of the cases of total blindness. Some say 15 to 20. So it's a very, very frequent cause of severe vision loss if not properly treated. And the treatments that we have today for uveitis are still not adequate. We are going into biologics, but it's often still prohibitively expensive. So it's a real struggle how to find the golden middle path. And I think that the more we know what makes it tick, the better therapeutic approaches we're going to be able to devise.
Absolutely. Reiko, is there anything else that you would want to cover and have people know about your research?
So we haven't actually identified the causative microbiome from the gut, and we are still searching for it to identify culprit bacteria. And yeah, and once we find it, maybe we can find a way to modify those in the intestine, or maybe probiotics treatment, some other methods to alter the immune responses to prevent uveitis or improve the symptoms, hopefully. Yeah.
Absolutely. We're looking for it very hard bioinformatically, and we already found some putative mimics, but having synthesized them and tried them out, whether they can induce uveitis, well, none of them were the peptide. Yeah. The results were not good enough to throw out, but not bad enough to throw out, but not good enough to follow. So we're still on the lookout. As databases are getting richer and richer, I hope that we are going to have more luck. And what Reiko hasn't told you about yet is her efforts with germ-free mice populated with human bacteria.
Ooh, could you tell us a little bit more?
Yeah, it's still preliminary, but we have collected the donor feces from human and healthy human, actually non-uveitis human, and we constituted the germ-free mice individually from the different donor. And we actually found out that all three lines, they do get certain level of spontaneous uveitis. So that means human microbiome can support the uveitis development. So as a next step, we would like to also test the uveitis patient flora and whether even it enhances more disease in this animal model. So this spontaneous model on the germ-free background will be a platform or something to model system to really hopefully identify and isolate the bacteria and also see how those community are involved, microbial communities are involved to really trigger the uveitis.
In the article that you had read, there were some reports of taking Behçet's disease patients and transferring them to mice, and the mice got sicker upon immunization. So that measures the adjuvant effect, not the antigen mimic, which you can only see that in a spontaneous disease model.
Fascinating. Okay. If our viewers want to research your work more, what would you suggest them searching on the internet?
Well, I have a lot of papers. If they just search for R.R. Caspi, they will find more than they want to read. They could search for Reiko Horai, because she publishes under Horai, and those will be focused mostly on the microbiome, but not only.
Brilliant, brilliant. Okay, well, thank you so much for taking the time to talk about these fantastic details that you're working with on a day-to-day basis.